In the second day of our international seminar on recent advancements in basic applied and material sciences, RABAMS 2025, we are going to start the technical session 6. In this session, we will have total 6 speakers back to back who will either join by online or will present physically. So, first speaker is Dr. Vivek Bandyopadhyay from Northwestern University, USA. Good morning and welcome to this seminar, Dr. Vivek Bandyopadhyay. We are very happy to have you as one of our resource person. And before going to start the session formally, I want to introduce you to the speakers. We are privileged to introduce Dr. Vivek Bandyopadhyay, postdoctoral researcher at the Center for Network Dynamics, Department of Physics and Astronomy, Northwestern University, USA. He earned his PhD in physics from the University of Bardon, where he worked on oscillation suppression and some symmetry breaking states in coupled quantum oscillators. His research interests span nonlinear dynamics, open quantum system, chimera states, and network of coupled quantum oscillators. Dr. Bandyopadhyay has published extensively in reputed journals, including Physical Review E, European Physics Plus, Chaos. So with this short introduction, now I want to invite our resource person, Dr. Vivek Bandyopadhyay, to start his valuable lecture. Vivek Bandyopadhyay, now his floor is for you.
Thank you very much. Thank you for this nice introduction. And I'm going to share my slides. Is it visible?
Yeah, it is.
Okay.
Okay, 1 minute, just take a picture of this one, slide on.
Can I start?
Yeah, screenshot. That's one. Okay?
Okay.
Okay, you can start.
Okay. That's it. Good morning, everyone. I am Mr. Vivek, and today I'm going to deliver a short talk on the topic of quantum manifestations of nonlinear dynamics, basically the quantum Schwarzschild oscillator as a test bed of this research. So all the natural oscillators are inherently nonlinear, and nonlinear dynamics is important to study the nature as it is without any linear approximation. And nonlinearity is crucial for self-sustained oscillations. Through nonlinear dynamics, you can uncover the principles behind self-sustained oscillations in natural systems. So these are two examples of nonlinear oscillators in our nature. This is electronic oscillator. This is human heart, which is a good example of biological oscillator. And this is the oscillation of the population density of rate predator model, which you can call an ecological oscillator. And you can see that the time periods can vary from milliseconds to years. There are many such examples of nonlinear oscillators in our nature. So, first of all, I'd like to introduce a classical self-sustained oscillator. So, what is self-sustained oscillator? First, we have to know that. So, this is the equation of classical self-sustained oscillator. It is a very good example. The name of this equation is classical Schwarzschild oscillator. And this part in this blue box is the amplitude equation of classical harmonic oscillator. So harmonic oscillator has a characteristic frequency but doesn't have any characteristic amplitude. So I would like to show you this video. So the amplitude of this harmonic oscillator depends from where we start. So if we start at a lower amplitude, it maintains the lower amplitude. If we start at a high amplitude, it maintains the high amplitude. So there are no characteristic amplitudes associated with harmonic oscillator. But if we add these two terms, this green term and the red term with the harmonic oscillator, it becomes the classical Schwarzschild oscillator, which is a self-sustained oscillator. So this term is called the linear gain, and this term is called the nonlinear loss. And nonlinearity is crucial here to let it be a self-sustained oscillator. So we can see this animation of self-sustained oscillator. So no matter where we start, it converges to a perfect and precise amplitude, which is one here after some time. So it has a characteristic amplitude along with the characteristic frequency. And you can see from this plot that here I plot the damping and the damping and pumping with the amplitude, and the role of the nonlinear damping is that because of this nonlinear nature of this red line, these two cuts at a point, and at this intersection point, we will get a stable amplitude. So this serves as the basis of extensive research in the classical nonlinear dynamics, this classical Schwarzschild oscillator. So now, for quantum investigations, we need a quantum analogy of that self-sustained oscillator. So what will be the form of quantum self-sustained oscillator? So we have seen that damping and pumping is necessary for the self-sustained oscillation in classical domain. So damping and pumping in quantum domain can be manifested by attaching quantum harmonic oscillator to the environment and allowing energy exchange. So here it is a schematic diagram of that system, quantum harmonic oscillator, and it is now attached with the environment, and it can talk to environment by exchanging energy. That is the crucial part. And this can be done using the formalism of open quantum system. That means quantum mechanics with dissipation, which is energy loss or gain. And by following this formalism, one can derive this master equation, which is the dynamical equation of density matrix rho. And this is now the mathematical form of quantum Schwarzschild oscillator. Here again, this equation in the blue box is the equation of quantum harmonic oscillator, and along with these two terms, which takes care of the interaction with the environment, it becomes the self-sustained oscillator. So this green term is the linear pumping rate, and the red term is the nonlinear damping term. And now, if we solve this quantum master equation, we will get a density matrix, which is a mathematical thing. So how to visualize that mathematical state in the phase space? So we need to convert this density matrix to a phase space variable, and here we employ this Wigner function, which is a function of phase space variable f t, and it can be calculated from this formula from this density matrix rho. And this is the probability distribution function as we are in the realm of quantum mechanics. So here, if we plot this Wigner function in the phase space, we will get a probability distribution. And from this distribution, you can see that the probability is maximum of this dark green. So here, we also get the limit cycle, or that limit cycle with the characteristic amplitude. But here it is the nature of this limit cycle is probabilistic, so it is not a distinct cycle. It is a fuzzy band-like grain. So this is indeed the dynamics of quantum Schwarzschild oscillator in phase space. Okay, so before going further, I would like to include three types of regimes in this research, which are called weak, deep, and extreme quantum regime. So weak quantum regime is where the rate of nonlinear damping is very much less than the linear pumping. So here damping is very low and pumping is high. In that case, the oscillator can populate much more energy level or much more level in the Fock space. This is the Fock space probability distribution, and you can see that the most probable state is away from the ground state. The peak is away from the ground state 0. And this gives this type of wave-like structure, which is the quantum form of limit cycle. And in this regime, the quantum results are comparable to the classical results. But if we make A greater than G, that means nonlinear damping rate dominates. And in that case, quantum noise becomes much more prominent. I mean, for that, the upper levels decay the ground state. And here, in this case, the ground state is the most populated state, and a few excited states are populated. Not very much states like the weak quantum case. And you can see that the ring is now becoming fuzzy than the ring in the weak quantum case. And it is the extreme quantum case where the k tends to infinite. That means nonlinear damping is at its best, and all the upper levels are decayed. Is there any question? And the nonlinear and all the upper levels are decayed to the ground state and the fast accelerated state. But still, we are getting a ring-like step which ensures that oscillation takes place. Okay, so classical emergent dynamics. Isolated oscillators are rarely found in nature. Networks of coupled oscillators give rich behavior, and the behavior is quite different from the behavior of isolated oscillators. And these are called emergent dynamics or collective dynamics of the network. And there are many different types of emergent dynamics. The main three are synchronization, oscillation suppression, and chimera. And these have been investigated rigorously in the classical literature. So first, I'll go through the classical inclusions of these three, and then I will move to the quantum counterpart of these. So synchronization. It is the adjustment of rhythms of oscillating objects due to their weak interest. So here it is a schematic diagram where two oscillators are coupled via a coupling channel, and this is the representative time series plot of the two oscillators. So they started, the blue one and orange one, they started from the different points, but due to their weak interaction, after some time, the trajectories overlap with each other. Gives rise to the complete synchronization. There are many examples of synchronizing in our natural life, in our nature, and one of the good example is the the blinking of the firefly in the South Asian rainforest in unison. So the fireflies are blinking together with equal rhythm. Next, the oscillation suppression state. So with interaction gives rise to synchronization, but what happens if the interaction is strong? So strong coupling can lead to oscillation quenching state at homogeneous fixed point, which is called amplitude dead state. So this is the synchronized state at coupling strength comparably low at 7.25. And if we increase the coupling parameter, this oscillation can be suppressed, and both the oscillator here populate the single fixed point, which is called homogeneous fixed point. So it is the state called amplitude dead state. And there is another kind of oscillation suppression state, which is called oscillation dead state (ODD). So with further increment of coupling, the amplitude dead state can undergo a symmetric breaking transition, gives rise to oscillation dead state. So here that single point splits into two, and now one oscillator is at this red point and another oscillator is at this blue point, it is at the orange. So this is another kind of emergent dynamics due to coupling. And the third one is most peculiar, which is called chimera. So it is an incongruous or counterintuitive coexistence of synchronous and asynchronous oscillations in a network of coupled identical oscillators. So the term chimera comes from the name of a creature Greek mythology, which with lion's head, goat's body, and snake's tail. So it is composed of different types of animals. In analogy to this name, here in this dynamical state chimera, there are different types of zones. So this is the instantaneous phase plot of a chimera state of a network of a self-oscillator. So the instantaneous stages of these oscillators and these oscillators are coherent, but the instantaneous stages of these oscillators are random at a single state, and this is called chimera state. Now, my main question is, what are the manifestations of these three property behaviors of classical domain in the quantum? So I asked the question, what is quantum synchronization? What is the manifestation of oscillation suppression in the quantum domain? And what is the quantum chimera? And we know that in the quantum realm there are many purely quantum effects or restrictions like Heisenberg uncertainty principle, discrete energy, or also inherent quantum mass, equivalence, etc. And for this, the manifestations of these collective behaviors may differ from the classical intuitions. So I will address all these questions one by one very briefly. So let us first start with quantum synchronization. So the minimum model to study the quantum synchronization is a quantum Schwartz-Lambda oscillator. This is the schematic diagram of the quantum Schwartz-Lambda oscillator. So with single boson gain and two boson loss, coupled to an external harmonic drive. So this is the master equation of this system where now there is an extra term called H drive, which takes care about the external drive, and this is the drive strength, and omega d is the drive frequency, and omega is the natural frequency of the oscillator. So in the frame rotating with the drive frequency omega d, this total Hamiltonian changes and looks like this one, where delta is the frequency detuning of the oscillator from the drive. So these are the results of quantum synchronization. Before going to the quantum results, I'd like to explain the classical one. So if a classical such oscillator is driven by external drive, then it perfectly locks itself with the drive phase. So you can see that the phase probability distribution shows a sharp peak delta function like this at a particular phase. So there is a perfect phase synchronization or phase locking of the oscillator with the drive. But what happens in the case of quantum system? So let us first take to the weak quantum system where the results are compared to the classical one, and we can see that the similar kind of phase probability distribution is showing a peak at nearly 1.5 pi, but it is not a sharp peak. It has a finite width. And in the Wigner function distribution, you can see that ring-like set is now lumped at this point of phase 3 pi by 2. So we can say that yes, indeed, a tendency towards phase locking is happening here due to the attachment of the external drive with the oscillator. But the phase locking is not strict, the peak is broadening. And as we move from weak quantum to deep quantum, this broadening increases, and to the extreme quantum, it increases more. So we can say that peaks are broadening in the quantum system, so no straight phase locking. So in this manner, the quantum synchronization deviates from the classical synchronization, and this happens because of the inherent quantum noise. We can increase the drive strength more and more to make the peaks narrow. But even if that the drive strength tends to infinite, we cannot get rid of the Heisenberg uncertainty principle and noise in that. So the peak will remain broadened, and that we cannot get delta from in unlike the classical. Now, the second one is quantum oscillation suppression. So there are two types of quantum oscillation suppression as I shown in the previous slides. The amplitude depth, the homogeneous state and fixed point, and the oscillation depth, the symmetry state and inhomogeneous fixed point. So first, let us investigate the homogeneous one. So for that, we have take two coupled Scotland oscillator underneath this is the amplitude equation where this is the coupling part, and this is the equation of the individual Scotland oscillator. And this classical oscillator shows two types of dynamics with the increasing coupling strength. At lower coupling strength, it shows oscillatory behavior, and at sufficiently higher coupling strength, it shows amplitude depth. You can see that both the oscillators, the blue ring and red dot, shows the x1 and x2 value. So here the oscillation is completely suppressed, and average amplitude of each oscillator is. But what happens if strong coupling can reduce amplitude depth in the quantum network also, which challenges the conventional understanding of the classical range. So this is the equivalent quantum equation. It must equation equivalent to this amplitude equation, and if we plot the equivalent measure is equivalent to the average square amplitude, here it is called a mean Boltzmann surface of A for the first oscillator. And if we stick to a particular Q1, Q is the mean strength, and here the Q1 was 0.2, so we stick to the point Q1 is 0.2, and we can see that as we move from low epsilon coupling strength to high epsilon coupling strength, the value of this is decreasing from 0 to dark. And at this 0 point, it is showing a ring-like behavior in the phase space, and in this dark point, it is showing this dot-like behavior in the phase space. And this is the manifestation of quantum amplitude depth. Though it is not a sharp dot like classical one, but it is quite like a lobe. centered at the 0,0 phase. So here we can say that in the case of quantum amplitude wave, the expectation value of A drive or A, which is equivalent to this measure, classical measure square amplitude average, tends to zero. You can see that here the value is not zero, approximately zero, but not perfectly. So this is the difference between the classical and quantum wave. So here the complete suppression of oscillation is not possible due to the quantum mechanics. And now the quantum oscillation test. For this, we take two terminals short and oscillator coupled under the conjugate coupling. This is the coupling which gives rise to the classical oscillation test. So you can see the classical dynamics here. So as we increase the classical, as we increase the coupling strength, we can go from AD state to OD state pitch for multiplication. And now is a good time to investigate quantum counterpart because we will be in OD state in the classical version. And this is the quantum master equation equivalent to the classical one. And here we also study the stochastic difference equation of the computer and noisy classical. What is noisy classical model? Noisy classical model can start can be a bridge between the classical and quantum. It is actually a classical model along with a noise curve which is equivalent to the noise of the quantum. So the results of the noisy classical model should match in the quantum model, and that will be a good check of the quantum. So these are the results of under the investigation of quantum oscillation, and here you can see that this is the quantum diagram. So here at low epsilon, the quantum oscillators are synchronized, showing oscillation. The ring light step in the phase shift. As we increase the epsilon, we will get a single lobe at the centered at the origin, which is the quantum amplitude deficit. But now, interestingly, as we increase the epsilon further, this single lobe splits into gives rise to the manifestation of quantum oscillation deficit, and we reported it first for the first time. And in a non-classical model, the results are actually supporting the quantum results. You can see that bimodal structure here at this epsilon 1.9. So I'd like to show you an animation for this whole transition scenario from this ring set. The coupling strength is increasing, and it is now at the unimodal step of quantum amplitude deficit, and now it is going to split into two lobes, gives rise to the quantum oscillation deficit. So this is the symmetric transition in quantum domain. And you can see that what is the difference of QOD and classical OD? So here the single oscillator shows two maximum probability waves. So this is the phase space dynamics of a single oscillator, and it can be here at this dot or here, together with half and half probability. That means equal maximum probability. So it is quite different from the classical intuitions because classically an oscillator cannot be at the two positions in the phase step at the same time. So this is an interesting step. It is like the superposition step of quantum mechanics. And here the oscillator shares two positions, and in this way the quantum oscillation deficit is different from the classical oscillation deficit. And now the last case is quantum chimera. To investigate the quantum chimera, we generally take a coupled ring of n identical quantum state and oscillator. So non-locally coupled. So what is non-local coupling? So this is a short network of 6 nodes you can see, and here D is 2. So D means the number of oscillators to which a single oscillator is coupled on either side. So here one is coupled to 2 oscillators, 2 and 3 on his right, and another 2 oscillators 6 and 5 on his left. So that is what it is true. So this is called a non-local coupling, where the coupling is not all to all, that is go up, and not local, that is only to the nearest group, in between these two. And to see the effect of quantum chimera, here the authors call it the quantum signature of chimera. It is not accurately quantum chimera, it is one type of signature of chimera. So we start from this type of initial conditions. So this is the initial conditions. These are the phase distribution of 50 oscillators. Here the network is of n equals 50, that means 50 oscillators, and D is 10 here. So for a particular number of oscillators, 21, the phases are almost coherent, and for the other patch, the other patch of the oscillators, the phases are incoherent. And we start from the with the in a coherent state centered at the phase end. So you can see that for I represented only the first two from the first batch of the oscillators and last two from the second batch of the oscillators to visualize this. So we can see that from the coherent batch, all the centers of the coherent, all the centers of the coherent states are similar. And for the last batch, the centers of the coherent states are different, randomly oriented on this side. And it is the initial condition, quantum initial condition, this one, delta t equals to 0. But as we move to delta t equals to 1, they squeezed with time. So they want to spread over this ring, and for that they started with squeezing, and we can see that the direction of squeezing are similar in this first batch, and direction of squeezing are different in this last batch. So this gives rise to the coherent zone of the first batch and the incoherent zone in the second batch or last batch. And this happens due to this coupling term to the Hamiltonian, and this is called non-local coupling in quantum network of quantum velocity. So here, the direction of squeezing defines quantum mechanics. So unlike the classical case where the phase, the amplitude, the center of the limit cycle, etc., define the chimeral state, here the direction of squeezing is defining the chimeral state. And it is in this way the quantum chimera is different from the classical one. And here also, it is coherent to the entanglement between two oscillators is higher than the entanglement between two oscillators from the incoherent zone. So with respect to, or in the light of entanglement, we can also differ this coherent and incoherent zone, which gives rise to the quantum signature of band. So these are the manifestations. Very briefly, I discussed manifestations of three main collective behaviors which are well established in classical domain, but these are the manifestations of them in the quantum. So takeaways and open challenges of this talk. I would like to mention three points. First one is genuine quantum effects. So we have seen that genuine quantum effects appear in the manifestation of classical collective dynamics in the quantum domain. For each of the three, I mentioned that the computational limit, it is the main challenge for the researchers who are working in this field. So it is hard to solve the system of large network. For a quantum network of n nodes, 2 to the power n-dimensional Hilbert space is required. And this needs huge CPU space and time for large Argentines. So this is the big challenge, and we are trying to overcome this challenge because we want to move from 2 oscillator network to 3 oscillator and then many oscillator network. And inherent quantum mechanical ideas. So there are different types of inherent quantum mechanical, absolute quantum mechanical ideas such as entanglement, infinite discord, etc. And these should have some connections with these collective behaviors, quantum synchronization, quantum symmetry, etc. So these need to be connected, and extensive study is needed in this direction. So I'd like to stop here, and if you have any questions, you can ask me or you can make any comment on my talk. Thank you very much for your attention.
Thank you so much, Dr. Vivek, for your lecture. Now, I would like to ask our audience or participants if they have any question. Ma'am, do you have any question? No. Okay, basically, I have a question, not questions, but I have some input because I am from chemistry background, but this is I whatever I thought I listened this is I think everything was done in a simulation process, no?
Yeah, everything is simulated here, and you know that experiment is quite costly in this quantum domain. But there are few proposed experimental setups like ion trap or superconducting circuit QED, and I would like to mention there is a recent paper which is still in the archive by a Chinese group who the authors in that paper presented the experimental proof of quantum ends to a Turner oscillator and its synchronization to the external, right? Very recently.
Okay. And anybody from online participants who want to ask any question? Hmm. If there are no questions, so I would like to thank especially from the organizing team of TPS College Patna for joining us. Thank you so much. Hope we will find you in different sessions also. Thank you so much. Best of wishes.
I would like to thank you also for inviting me.
Thank you.
Thank you. Thank you very much.
Okay, bye. So in this technical session, we will have our second lecture will be by Dr. Manish Devnath from CSIR Indian Institute of Chemical Biology, Kolkata. Dr. Manish, are you there?
Yeah, good morning. Yeah, good morning.
Yeah.
Very nice to see you
Yeah.
after a long time.
Yeah. So how are you? Fine? Yeah, good. Okay. First I want to introduce yourself and then you can start your presentation. Okay?
Okay, actually, am I audible?
Yeah, you are audible. I think. Yeah, audible. Okay, no problem.
Okay, so let me load my slides.
Yeah. Please.
We are going to start the presentation right now.
Yeah. Okay. Start it.
So we are going to start it now, right?
Yeah. It will be continuing.
Okay. Okay. So
Okay, Dr. Manish Devnath, welcome to RABAMS 2025 to this international seminar. We are in the second day. Now formally I want to introduce. We are honored to welcome Dr. Manish Devnath, scientist at the CSIR Indian Institute of Chemical Biology, Kolkata in the Organic and Medicinal Chemistry Division. He earned his PhD in Chemical Biology from Indian Association for the Cultivation of Science, specialization in nucleic acid research. Dr. Devnath has published in leading journals including Nature Communication, Anglo Entertainment, Nucleic Acid Research. He is also recipient of prestigious international fellowships such as Lundbeck Foundation Postdoctoral Fellowship from Denmark, JSPS Fellowship from Japan. His current research integrates artificial intelligence and machine learning with molecular biology to advance drug discovery and diagnosis. With this short introduction, I am now to hand over to Dr. Manish Devnath. You can start.
Dr. Paluti, my slides are visible.
It is visible.
Okay. Thank you for the introduction, and I really like to talk about my research. And thank you, organizers, for giving me the opportunity to speak about my research. As Dr. Paluti already introduced, I have started my lab here at IICT in the Organic and Medicinal Chemistry Division in 2022 August, and it has been approximately 3 years. So here we have been trying to do some new things, and so my group basically is trying to use artificial intelligence like machine learning and deep learning just to solve biological problems. So the problems which we are focused on is development of AMPs like antimicrobial peptides to combat antimicrobial resistance. We are trying to develop a drug conjugates to combat a disease like cancer, and then we are also working on development of nucleic acid TPS cameras which can be used as biosensors. We are also working on the development of bioink, which can be used in bioprinters for constructing 3D printed organoids or 3D tissues. So based on this short introduction, today I focus on this AMP part. So this is one of the main focus of my lab. So today I will be discussing this development of AMPs using AI-based techniques which we have recently done in our lab. So the challenge in the this field of research is the antimicrobial resistance, which is developing very rapidly and one of the major problems in India as well as over the world because about 3 million.
Dr. Manish, actually, I think you have to share your entire screen actually. Maybe that's why it is not moving actually. Slides are not moving. Okay, now it's okay. Okay. You are
sliding it.
Okay.
Slowly.
Go to.
Just a second. Should I share it again?
Yeah, share entire screen actually. Then it will work, I think. Yeah, it's okay. I think share entire screen. Yeah, now it will be fine. Just do it in presentation mode.
Now,
Okay, fine. Fine.
okay. So as I was saying that we are using AI-based techniques to solve different biological problems in developing antimicrobial peptides, development of bioink for 3D printed tissues, development of nucleic acid isomers for developing biological sensors. So today I will be speaking on development of AMPs, which we have done recently using artificial intelligence tools. So the major challenge in this field is the antimicrobial resistance. What is antimicrobial resistance? The bacteria, we are prevalently infected by bacteria, different sort of bacteria, and they get resistant over a period of time. So whenever we are getting infected with the bacteria, we take antibiotics, and after some time the bacteria evolves. It gets resistant towards the antibiotics. So this is a major challenge in India as well as globally, as about more than 3 million people are affected by drug-resistant infections, which is as per the report of CDC 2019, and more than like 3.5 lakhs deaths per year worldwide are happening due to multi-drug resistant bacteria. And there are different pathogens which are in need to pathogen list like the penicillin-resistant staphylococcus and then the Pseudomonas aeruginosa, Acinetobacter baumannii. These type of strains are increasingly getting resistance, and our last resort antibiotics like penicillin and different kind of antibiotics are getting dried up. So antibiotic discovery pipeline is increasingly drying up, and we need new novel, stable, potent, safe alternatives to conventional antibiotics. So at this point, our group is trying to develop new antimicrobial peptides. We are presenting AMPs as new alternatives against the conventional antibiotics. Although work on antimicrobial peptides have been done from like long back, we are trying to develop some of the modified antimicrobial peptides which can be a alternative to the conventional antibiotics. So why antimicrobial peptides? Because it has a broad spectrum efficacy, membrane disrupting mechanisms, and it is less likely to develop microbial resistance compared to the conventional antibiotics. Already there are few gene-based antibiotics in the market like colistin, vancomycin. Like again, this structure is mirapavidin. It is in the clinical trial. Daptomycin also in the clinical trial. So our idea of developing AMPs is derived from animal host ecosystem. So all the animals like insects, human beings, frog, wasp, snake, they have inherently some host defense mechanism, which is part of their innate immune system. So these peptides actually exhibit potential antimicrobial activity and naturally protect our body. So what we are trying to do, we are trying to do the genome mining, proteome mining, and then mine these peptides and try to get inspired from the nature and develop new antimicrobial peptides based on these host against proteins. Okay, so again, I would like to add that modification of these peptides, like already done with cathelicidine and temporine, cathelicidine found in snakes, temporine found in frogs. People have developed cyclized analogs and they have shown increased stability and antimicrobial potency. So why this step-built antimicrobial peptides we are talking about? Because as we know that most of the peptides, these antimicrobial peptides and post-fitness peptides, they are ill peptides. And they are susceptible to proteolysis by proteases and peptidases of our body. Although there are some of the peptides like AL37 and cathelicidine which are ill peptides, but they have shown good property in the biological system. But we still want to develop stable peptides which have much higher target specificity, binding affinity, and improved stability. And it has enhanced antimicrobial potency, like about 100x more than the linear tail peptides. It has a reduced cytotoxicity for the normal human cells. And then there are known examples which I have already talked about, like here 12 encoding analogs, cathelicidines. So this kind of stapling, what stapling means is basically this kind of conjugation, this kind of linkage between two amino acids. It may not be end-to-end, it can be end-to-end cyclization, or it can be somewhere in the middle. So this type of conjugation it actually increases the stability, affinity, and specificity of this peptides. So this is called stable peptides. So now, where does the AI/ML come in? So why we use AI? The development of peptide-based antimicrobial peptides is like laborious. Like see, if we want to develop like 10,000 or 1,000, even 1,000 peptides, we want to synthesize and check their biological properties. It will take a lot of manpower and resources and time. So we want to come up with something quicker, so that the pipeline goes from months to days. So AI/ML is a little bit advantageous in this matter. So people have been also using the computational based drug discovery methods, which mainly looks at the binding pocket or the target structure, and based on the structure of the binding pocket, which develops molecules. So why AI/ML? So compared to the computational resources, static computational resources, AI/ML actually gets trained by already known data. So whatever data on peptides which we have on the databases that can be collected, that can be curated, into a form which can be read by the machine and the computer, and then we get our system trained. And after by descriptors, like if we are doing the traditional computational analysis, it can only find molecules based on the chemical space or the binding pockets. AI/ML on the other hand can work on like 200, 500, even 1,000 descriptors. So much more efficacy is there, and it can also predict modification pattern for maximum activity. So there are a lot of ML-based tools already available, like random forest, XGBoost, support vector machine, artificial neural networks are there. But in our lab, we are working on developing a deep learning based system, which is customized, which is not the traditional algorithm which we have developed in house, and we are training it continuously, and we are developing new methods. So it considers many features which is based on sequence stability, already known MIC values, structural motifs, and the chemical space, immolating activity. All these type of features are considered in this. Our research objectives mainly, yes, our research objectives mainly go like that. Like, if you take an example, we curate a data set. We curate a data set of about 10,000 post-sequence peptides, and then we screen them using our in-house developed deep learning. So once we screen them, we can get about 100 prioritized candidates, which we synthesize them by solid phase peptide synthesis, and then we do the scaling. And then the stable peptides are further evaluated for their toxicity, stability, antibacterial activity in vitro system. We have some clinical collaborators on where we take the resistant pathogens, bacteria, and then we test them in vitro. And then we also, as I mentioned, that we are working on 3D tissue models. So we have these tissue models and we infect them with the bacteria and then we test our molecules in 3D tissue models. So the pipeline goes like this. We collect the already known peptides from the peptide database. Like there are a lot of databases, TPS, TPS, and from those resources we collect the sequences. We do the feature extraction. Now all the features are important, so we then again do a feature selection and find out the relevant features. Like suppose we are selecting about 500 features, and then using those features to train our system. And after training the untrained model becomes the trained model, and the trained model, if you can complete the query sequence, then you can report that whether the peptide has an antimicrobial activity, hemolytic activity, and other properties it can report. And we can screen about 10,000 peptides to 50 peptides in about 100 minutes with more than 95% accuracy. So this is what our data set look like, and this graph actually indicates the peptides which we have used to train our machine learning systems. So these are the number of peptides against particular bacteria, and these are all gram-negative bacteria. These are all gram-positive bacteria, and these are all peptides which have hemolytic activities or non-hemolytic activities. So these are all the numbers of peptides which are activating the particular bacteria, gram-negative, gram-positive, and the hemolytic properties of the peptides. So mostly the human, in case of hemolytic, we have taken the human data on human data. So the development of ML system, how do we develop the system? So once we get all the data, all the peptide sequences from the databases, we do this type of mapping. And after the mapping, these clusters actually indicate a similar the peptides having similar chemical space or similar properties. And from these clusters, these peptide sequences, we extract the embeddings or the features or even say descriptors. This is the main thing, and then we use this descriptor to train our system. So for our deep learning system, it is based on language modeling. Language modeling you must be very familiar because we use language modeling regularly in form of ChatGPT or Gemini. These are all language modeling systems. So this is our very new protein-based language modeling, ProdBERT. It is called ProdBERT, and this ProdBERT actually takes up all these features, our descriptors, and it is trained, and then we can predict the high, medium, or low. We can do three type of classification of the antimicrobial effects on the efficacy, high, medium, or low, and we can also do the binary classification like high and low activity effects. So next, what we have done is we have checked whether our in-house developed deep learning system is have more efficacy than the already available machine learning algorithms. So we have checked like four different algorithms: support vector machine, XGBoost, CatBoost, and then our team model. So among them, you can see that this black one is the our tuned model, and if you see the true positive rate of this tuned model is higher than the other other algorithms. You will see the data for the gram positive and the gram negative bacteria. Since the data set for the gram negative is little bit larger than the gram positive, so for the gram negative data, you can see that true positive rates are much higher, the difference is much smaller, and false positive rate is much lower. So again, we have also checked for the binary. This is for the three classification models like high, medium, and low activity, and this for the binary classification, high or low activity. So again, we can see that this deep brown one, this is the human model, and it has much more higher positive rates, and it indicates that it has more capability to give exact data. It has higher accuracy compared to the other algorithms. So again, apart from these curves are actually, if we go into the technical details, these are called ROC curve, area under curve. So these ROC curves actually indicate the accuracy of the algorithm. So apart from the ROC curves, we can use, there are several other metrics which can report on the performance of different algorithms like precision, matrix, coefficient. These are all numerical terms, and which indicates the accuracy of or efficacy of the neural system. So you can see that is the brown one. This brown one you can see. So this has higher accuracy, and all the parameters are higher, and Hamming loss, which is lower for this tuned one. So this all metrics, all 7 metrics, this indicates along with the performance of that our in-house developed neural system or real system is much superior than random forest, X boost, cat boost, and SVN. What are the other neural systems we have investigated? So our pipeline goes like this. We have taken different library from both the types, and about the library size is about 10,000 sequences, and then we have this gram negative and gram positive classifiers. As you can see from our podcast, this classifier can act as a gram negative or gram positive classifier, means it can show the efficacy of the peptides against gram negative or gram positive organisms. So from that, we shortlist some of the peptides, and then the hemolysis classifier that whether it is showing hemolytic activity or not. So we can shortlist some of the peptides, and then the peptides are actually synthesized. So this graph here shows the consistency of the different peptide libraries against gram positive, gram negative hemolysis with our this library which we have taken to study. So this is the changing property that is hydropathy, isolated points, and molecular weight. So this shows the distinction of library which we have taken. And these are some of the sequences which we have investigated. There are a lot of sequences, like thousands and thousands of sequences, and we are regularly investigating it with our ML system and updating our system with this data. We are feeding again with this data, which is called reinforcement learning, which is a common practice in ML. So whatever data we are getting every day, we are again feeding into the system. So our system accuracy is increasing regularly. So this is some of the peptides which we have shortlisted for biological investigation, and if we try to see the entire chemical space, so these are the areas from where we have chosen our peptides for biological investigation. And from the structural analysis, you can see these spectroscopy, we have seen that all the peptides which are showing efficacy in the ML-based studies have a different structure. They are not random coil structure. They have something. They are beta sheet or they are. So then we have collaborated with we are getting that samples from there. So we got registered strains and we checked the efficacy of the peptides, the synthesized peptides against all the strains. SA is the Staphylococcus is the non-resistant one. This is the penicillin-resistant Staphylococcus, and this is the aeruginosa, which is one of the major concern and the priority pathogen and the most important pathogen at the point, and people are trying to develop antibiotics against. So we found that our among all the peptides, these two molecules these are the most effective ones and have considerably less hemolytic activity compared to the current post-genus peptides. One more thing, one more part of the information in our work is carrying post-genus peptides are very long, like 20 to 40 amino acids. Our peptides are much shorter, like about 6 to 9 amino acids. So if we reduce the size of the peptides through some optimization, it gets easier to synthesize and easier to cellular penetration studies and all those things are much easier for larger molecules are so this is a study which shows that after treatment, the bacterial cells are increasingly getting life disrupted after treatment of increase. And then this is just data which shows our study with 3D tissue models. We have developed 3D tissue model using epithelial cells, skin epithelial cells, and also the lung epithelial cells. So this one is skin epithelial cells, and we have developed this model treated it with bacteria. After 15 hours, the bacteria gets the bacterial load is verbal and you cannot see the epithelial boundary here. After treatment for the antimicrobial peptide for about 24 or 25 hours, this bacterial infection gets cleared up. And after 35 hours, there is substantial amount of recovery. You can also see the epithelial boundary again, and the efficacy of has been much higher than the ill and other peptides also stated, and the efficacy also was checked with commercially available peptides like the drugs like vancomycin and colistin, and the efficacy was on par. But compared to the vancomycin and the vancomycin and colistin, our molecules are much easier to synthesize. So it has some of the. This is another data which I have been talking about. Lung epithelial cell tissue model, and we have treated it with aeruginosa because aeruginosa mostly infected the lung cells. So in this, we infected it with aeruginosa, the lung epithelial cells. We have checked the efficacy after 35 hours. Epithelial boundary here, bacterial infection gets much cleared after 35 hours, and these are my students who have done predominantly work. Who has done mostly the AIML part, Shobhi, who has done some of the synthetic and biological studies. Seva is working in my lab, so she has optimized this peptide synthesis and cyclization methodologies. So, in conclusion, our email-based peptide design, pipeline enables rapid discovery of hot, stable non-colloidal entities. Although we are constantly improving, and at this point, our efficacy of the system is about 95%. We are trying to improve the databases, and as the size of the database will increase, the accuracy and efficacy of the system will increase. And next, we are looking to develop peptides which are specific to gram-negative, gram-positive, or even specific to a particular or type of bacteria. So we train the data, train the algorithm with only peptides against that particular bacteria like Aerogeosa, and then we develop some of the new peptides with modification and the further investigation. So this is a kind of closed loop system that we are doing AI/ML studies, and then we are also doing the biosynthesis and biological investigation, and hoping to find out some new interventions. So thank you. At this point, I would like to stop and this is my group, very small group, and some of my collaborators, and most of my funding from SAR, AMRS, CSIR, and ISIR.
Thank you, Dr. Manis, for a wonderful lecture. And now I would like to first I will take some questions from online forum. Kumari Josna, do you have any questions? Any other on?
Hello, Dr. Yeah, Dr. Dirna.
Yeah.
Oh, yeah, very nice talk. So I have just a simple question. So, during using the ML method, the machine learning method, so yeah, for the peptide things. So the thing is that sometimes overfitting things come into the picture. So how do you compare with that or your methods? So you are using the database to train the you are using the ML method to train the database. Right? So sometimes the overfitting
We are
things.
using data sets to train the MLs. Okay, anyway. Yeah.
So I just want to know how you means manage the overfitting things. You know, there are large number of database. I think I guess that there should be.
Yes.
So that thing comes into the picture, particularly when on the peptide things of homologous sequences are there. So,
Yes, it is a very laborious process. We need to curate the data set from databases. We have to take out all the peptides, and then we have to manually curate. So that is one of the most laborious things to do. And we have to see whether the peptides are getting repeated or some of the, like you said, the sequences are there. So we have to do some of the sorting of the peptides, and we have to make it in a uniform way. The data set has to be carefully curated so that the overfitting problem does not arise. So yeah, that is one of the challenging things we are.
very nice. Thank you.
Thank you.
Thank you. Now, is there any question from the audience? Yeah, Dr. Sandeepa and
Dr. Gurna, it was a fantastic talk. I wanted to know how do you establish connection between your machine learning data and experimental findings?
Sorry, I cannot hear you.
I wanted to know, did you check the validation of your machine learning data with the experimental one?
Yes. That is the most. As I was saying, our lab is working on a like closed loop system. First, we are using AI analysis system to screen peptides, virtually screen peptides, and then we are synthesizing them, and then we are also making the analogs, the stapled analogs, and then we are checking them in the biological system. Because we are not only checking the deep molecules, we are also checking some of the paired cases, and that is how we are determining the accuracy. There is actually 2-3 points of validation we do in machine learning. First, we divide the data set into two parts. That is the training part and the testing part. Suppose the entire data set is 8,000, and then we divide it 8,000 and 2,000. So what happens is we train it with 8,000 data set and validate it with 2,000 remaining test data. So this is the primary part of validation. And secondary sort of validation comes from the wet lab validation, which we do where we really synthesize the molecules and test them in the biological system.
thank you, ma'am, and thank you, Manish, Dr. Manish, for and formally, I would like to thank you for joining us on time, and hope to see you soon. Thank you. Bye bye.
It's very nice to see all of you.
Thank you. Thank you. Our next speaker of this technical session is Dr. Vaishmani Sharma from IIT BHU. Dr. Sharma, are you there?
Yeah, can you hear me?
Yeah, I can hear you. Yeah. Good morning and welcome to this international seminar RABAMS 2025 in TPS College Patna through online forum.